Cells are the fundamental units of life. They are complex and dynamic structures enclosed by a thin, flexible barrier called the plasma membrane. This membrane is not static. It behaves more like a fluid mosaic. This fluid-like nature of the membrane, known as membrane fluidity, is crucial for cell survival. Membrane fluidity refers to the ability of lipids and proteins within the cell membrane to move freely. Imagine a sea of lipids with proteins embedded within it, constantly shifting and rearranging themselves. This dynamic movement is essential for various cellular processes. Without fluidity, the membrane would solidify, hindering essential functions like nutrient transport, signal transduction, and cell division. In essence, membrane fluidity is vital for maintaining the integrity and functionality of the cell. Temperature plays a significant role in influencing membrane fluidity. Think of butter. When cold, it's solid and rigid, but when heated, it becomes soft and fluid. Similarly, cell membranes become more fluid at higher temperatures and more rigid at lower temperatures. Why does this happen? At high temperatures, the increased kinetic energy causes the lipid molecules to move faster and more freely, increasing fluidity. Conversely, at low temperatures, the lipid molecules pack tightly together, restricting their movement and making the membrane more rigid. This temperature dependence of membrane fluidity has important implications for organisms living in different environments. For instance, bacteria living in hot springs have membranes with a different lipid composition compared to bacteria living in the Arctic, allowing them to maintain optimal fluidity at their respective temperatures. Besides temperature, the composition of lipids in the membrane also significantly influences its fluidity. The membrane is primarily composed of phospholipids, which have a hydrophilic or water-loving head and two hydrophobic or water-fearing tails. The length and saturation of these fatty acid tails play a crucial role in determining membrane fluidity. Shorter fatty acid chains and unsaturated fatty acids, which have double bonds in their tails, increase membrane fluidity. The kinks introduced by the double bonds prevent the phospholipids from packing tightly, keeping the membrane more fluid. Conversely, longer fatty acid chains and saturated fatty acids, which lack double bonds, decrease membrane fluidity, leading to a more rigid structure. The cell can adjust the composition of its membrane lipids to maintain fluidity under different conditions. For example, at lower temperatures, the cell can increase the proportion of unsaturated fatty acids in the membrane to prevent it from becoming too rigid. Organisms have evolved remarkable ways to regulate membrane fluidity in response to environmental changes, particularly temperature fluctuations. This adaptation is crucial for survival as it allows them to maintain optimal membrane function across a range of temperatures. One example is the change in membrane lipid composition observed in extremophiles, organisms that thrive in extreme environments. Bacteria living in hot springs have membranes enriched with saturated fatty acids, which help maintain membrane integrity at high temperatures. In contrast, fish living in icy waters have membranes rich in unsaturated fatty acids, preventing them from solidifying at low temperatures. Another example is hibernation in animals. During hibernation, animals experience a significant drop in body temperature. To cope with this change, their bodies adjust the lipid composition of their cell membranes, increasing the proportion of unsaturated fatty acids to maintain fluidity even at lower temperatures. These examples highlight the remarkable ability of organisms to adapt their membrane composition and maintain fluidity in response to environmental challenges, emphasizing the critical role of membrane fluidity in sustaining life.